So I took the title of my talk from this movie, which came out in 2004, called Super Size Me. How many of you in the audience like eating McDonald's? It's nothing to be ashamed of, but okay. So Spurlock here enjoyed eating McDonald's too. And he decided to do a small investigation of his own on the effects of a McDonald's diet on his body. And around this time in McDonald's, at least in the US, they had this phenomenon called supersize me, which is you could go to the McDonald's, you could order your Happy Meal or whatever, and you could, for the same cost, supersize it. So you could get double the amount of fries, double the amount of Coke, double the amount of meat in your burger, right? And so he took this on as a challenge, and for 30 days, he ate McDonald's morning, noon, and night. Now, admittedly, this is not the most, it's not the easiest diet to follow, right? And after 30 days, there were lots of very uncomfortable things that happened to him. If you get time to watch the documentary, I would encourage you to do that, including putting on 12 kgs in 30 days. He also reported mood swings, depression, anxiety, and really the reason he put himself through this experiment was sociological. His argument was that the economy behind fast food is that you want to get people hooked to this food, and a number of people who are not capable of making their own food or want a quick fix would eat McDonald's. And he wanted to make the claim that having this supersize phenomenon was not really good in terms of public health. And following this is this report that came out from the Institute for Health Metrics and Evaluation, which basically shows that obesity is on the rise. And that's not going to be breaking news for anybody in this audience. What is interesting, of course, is that India is broken into the top 10 rank as far as this is concerned. We're number 3, and we're probably going to get better at this statistic. On the contrary, though, adult obesity is increasing, but if you look at weight as a parameter, and this is a report that came out in Lancet 3 months ago, the number of adolescents who are underweight is also very high in this country. I mean, if you look at this particular picture of the world, look at this. India is like bright red here, and it's really dark here, right? So it's really a puzzle. How are these underweight adolescents turning into really fat uncles and aunties, right? What's going on here? There's obviously a biological phenomenon here. Everybody is not surviving on a McDonald's diet, right? There must be something else at play. So I'm going to take this to a slightly different way of thinking. A hypothesis was put forward by David Barker in 2002. It's the Dohat Hypothesis, the developmental origins of adult health and disease. And this hypothesis predicts that if, as a child, in the fetal or during early childhood, if you experience suboptimal nutritional conditions or stresses, you are likely to develop non-communicable diseases later in life. And non-communicable lifestyle diseases includes things like obesity, diabetes, hypertension. So clearly what is happening is that for us as Indians, we may have experienced a term early in our childhood, or maybe our grandparents or parents might have experienced a period of low nutritional conditions, bearing which if we now or I now go and eat McDonald's, predisposes me to put on weight much faster than somebody else, right? And the question I have is, can we study this phenomenon in flies? So can the Drosophila model system be adapted to study nutrition and metabolism. I mean, how different are we from the fly? Are we different from flies? Right. I mean, we can't fly, which I think is really the biggest thing, biggest crib I have about not being like a fly. But you know, we can grow. So if I become fat, I'm most likely going to put on a lot of abdominal fat, and then I'm just going to be twice my size, right? Whereas fly, they have an exoskeleton, and they can't really grow, right? Once you are born of a certain length and height and diameter, because the skeleton is on the outside, you can't really grow. So do you think a fat fly is possible? I hear one no. But luckily, I have a job because the answer is yes. So flies can be fat. And although their external skeletal apparatus is not going to expand to a great degree, they do put on triacylglycerides, or the fats that you and I have. Much of the biochemistry in terms of lipids, proteins, and sugars happens to be very similar between us and flies. So we also have a diabetic fly. There's a very interesting experiment done more than 15 years ago, where this particular group, it was actually Kim Rulifson in Stanford, identified a bunch of neurons which they say makes glucagon-like hormone. For the insect, it's adipokinetic hormone. And what they could do, because of Drosophila genetics, is just ablate those neurons. They just got rid of them. And they found that the fly actually had higher amounts of sugar in its hemolymph, or in its blood. And now the field has moved on from this quite dramatically, and it is quite clear that both obesity and diabetes are phenomena that we can easily reproduce in the fly. My interest is in malnourishment, and I think that I have a model that recapitulates malnourishment in flies. So here is a fly that has been fed food in the laboratory under laboratory conditions for the entire duration of its development. And here is a fly who, for a very short period during its development, was denied food altogether. Flies eat in the lab sugar, yeast, corn flour, dash of agar. And what I did here was basically take the larvae and take away everything except a little bit of sugar, allowed the fly to complete its development, and lo and behold, I get a fly that is malnourished, or in public health parlance, stunted. So this is the life cycle of a fly, and it is during this period that I am withdrawing nutrition from the fly. I will also use the word nutritional stress during my talk. And what I'm really focused on in my postdoc is asking the question that if I subject a larva to nutritional stress during this last little bit of development, how does it survive it? I'm still getting pupae, and I'm definitely getting adults. So what are the mechanisms? If you look around us in India, we are ranked number 1 for malnourished children, right? But when you look at malnourished children, you'll find that all their organs and systems, at least morphologically, look the same. If you're deprived of nutrition during childhood, during fetal development, it's not like you're going to get one finger less, right? The whole body will proportionately decrease. So there must be some mechanisms that might be in play in order to make sure that despite poor nutritional conditions, you're able to complete development. And I'm sort of interested in understanding what those mechanisms are. I'm going to tell you 2 stories. This is the work that I've done in my postdoc. I'm not going to go into the technical details. If you'd like to know more about that, come and talk to me at the break time. So the first question was that during development, if you don't get adequate nutrition, what happens to neurons, right? And the second question was particular set of neurons which make hormones. I mean, a lot of people here asked questions about hormones. So I was particularly interested in neurons that make hormones and wanted to understand if I could find hormones that allow an organism to complete development under nutritional stress. I said, all organs are not treated equally, even though in terms of size, that's what we observe. There's a phenomenon called brain sparing, where if you look, and this was coined by pediatricians in, just move ahead, who were looking at children who were born smaller than normal. And what they found is that the brains of these children did not scale down to the same extent as perhaps the length of the baby or the digits themselves. And this idea, this was in 1970s, was that somehow the brain can compensate for nutritional loss. And a very elegant study in Drosophila in 2012 basically showed that when you deprive larvae of nutrition during development, glands, the organs that are not really that important, for example, the salivary gland, really reduce in size, whereas organs that are important for the final fly, such as the brain, the CNS, continue to grow. So flies also show brain sparing, sorry. I focused on a particular set of neurons in the fly brain called neuroendocrine cells. The larval brain has about 100,000 neurons, and those that make particular neurohormones have a transcription factor called DIM, and there are about 300 of those. And they're spread all over the larval brain. And I'll summarize the entire story that I have with that particular investigation thus. So what I found is that in a brain that is starving, neuroendocrine cells need to continue to make proteins. So although the whole body is experiencing nutritional starvation, there are some cells that need to continue to make protein because they are important for you to survive. So they can't just shut down. Neuroendocrine cells are one such set of cells. The second thing I found was that when you turn off signaling pathway in these cells, which is driven by nutrients, and that's the insulin signaling pathway, another pathway has to come into play, and that pathway is driven by intracellular calcium signaling. So it's a way for cells that are deprived of nutritional signal to continue to make proteins in the absence of any external sources of protein. That's the sort of the first story. What this has really led me to do, and this is something that I'm particularly excited about working in Drosophila with, is that I have been able to go from organism research to tissue and finally to cell. So I'm able to go take a very nice resolution all along and come up with an organismal biology approach. And this is what was very different between my PhD thesis and what I'm doing now. In my PhD, I had a test tube and I could change whatever parameters I wanted with lipids in a test tube and ask certain questions. They were fundamental, and I still think lipids are amazing. However, here I am basically doing some perturbations at the level of cells, but I'm seeing some phenomenology which is at the levels of the organism. The second story that I have is focused on these neuropeptides themselves. I find them particularly exciting molecules to work with, simply because unlike neurotransmitters, dopamine, serotonin, which are released by our neurons, neuropeptides act very slowly. They don't act as fast as neurotransmitters. They're slow and steady, and their function is more to do with modulation. So for behaviors such as aggression, mating, even physiology, food, insulin, all these have a much longer timeline of activation or activity as opposed to neurotransmitters. And that's what makes them really exciting for me to study them. So the next question I asked in my project was, if there are 300 neurons that are responsible for survival of the larvae under nutritional stress, can I figure out one particular or few neuropeptides themselves that are important for this process? There are over 40 neuropeptides that have been described for Drosophila. A number of these neuropeptides are conserved with mammals. Just in this little brain lobe itself, you can see about 20 spots, and each one of these spots is a different neuropeptide. So here is the power that Dr. Ranganath was talking about, the power of Drosophila biology. where you can now use genetics to drill down and ask a cellular question. So what I have done is just summarized a lot of work using good old-fashioned genetics and some really funky tools that we have in Drosophila, figured out which neuropeptides are important for this process. This is just basically a UAS GAL4 screen. It's a genetic screen that we carry out to ask a particular question. In my case, what neuropeptide is important for survival under nutritional stress. This is basically a tool where you overexpress a protein that makes GFP, and the GFP only comes on in neurons that are active. So if this is the larval brain on top here, you can see only 5 neurons that came on to be active. And I was able to then figure out from some other methods what these 5 neurons are making. And they made 2 particular neuropeptides, SNPF and corazonin. And then so going back from this experiment, I was able to validate genetically that these 2 neuropeptides are important. So again, in this project, I'm very excited that I was able to go from something at the organism scale to down to finding out cells that are responsible and to the actual neuropeptides. And the last question is something that I'm still trying to figure out. Getty's lab works on intracellular calcium signaling. So we're trying to figure out how does intracellular calcium signaling affect these particular neuropeptides. And this is particularly interesting because in neurons, calcium can come from very many different sources. First, of course, is the fact that when your neurons fire, a lot of calcium comes into the cell. And those are all voltage-gated calcium channels. But sitting right here inside the cell, in the endoplasmic reticulum, is a storehouse of calcium. So the concentration in the ER is about 1,000-fold larger than it is in the cytosol. So in Getty's lab, we've been trying to figure out if neurons have so many different sources of calcium, what is the point of having this storehouse of calcium? And is there any modulation that this particular calcium, that's why it's called intracellular calcium, doing to the neuron itself and regulating its function? So this is now just a thought exercise for me. How does calcium affect neurohormones? And this is the way I'm thinking about the problem. Either calcium can do something to the mRNA level, or it can do something to the protein level, or to the vesicles, or finally to the release. And I've more or less eliminated the RNA levels. I've looked at the peptide levels with, again, an antibody. All these are tools that I'm just trying to show you to illustrate that you can go from the organism, whether it survives on sucrose, to the cellular biology quite quickly in Drosophila because of the toolkit that we have. And then finally, I want to just show you a movie where I feel that that it's not transcription. I think it might be peptide levels, but I also want to test for release. And what I can do in the fly system is I can dissect out the brain and put it under a microscope, and I can actually monitor the activity of the neuron under the microscope. And this is just an experiment to show you how that looks. So at the red part is where I have added potassium chloride, 70 millimolar potassium chloride, and I've activated the neuron, and I'm able to follow the kinetics of what's happening inside the cell body because of this genetically encoded tag that I have. So this is the point where I am at right now. I'm trying to finish up, trying to answer this question that I have 2 neuropeptides that are involved. One of them actually is an analog of gonadotropin-releasing hormones in mammals. The other one is an analog of neuropeptide Y in mammals. So I have 2 neuropeptides, which I think are important for development under nutritional stress. And I'm trying to figure out the cellular mechanism of how they work in context of intracellular calcium. And that, I would say, is the power of fly biology. And finally, I started out with this Dohat hypothesis. I think the fly model that we have is very powerful to try and answer some of the mechanistic questions of the Dohat hypothesis. So what happens in public health and clinical studies is that if you have experienced early life nutritional stress, and then later on you have come to an environment where you get high calorie food, sufficient food, enriched environment, then what finally happens to you at 35 is very different from if you had a persistent chronic malnourishment throughout, right? And this in public health is called confounding effects. And it also in the cases of, say, education. So you would expect that person who has experienced poor nutritional conditions, suboptimal brain development, and will have cognitive defects. But it turns out that if you provide 20 years of tuition classes, you more or less not have any cognitive defects when tested for at 25. So clearly there are other factors at play, and the brain is quite plastic. But I think Drosophila is a great model system to ask a very simple question. Okay, I had poor nutrition during my fetal development. What impact did it have to my brain structure and to my physiology? And this is a very particular problem for physicians because there is a condition called failure to thrive. So despite interventions, there are some children who will never recover from the nutritional insult that they got during fetal development. Why does this happen? We don't know. So can Drosophila be a model to try and understand maybe some molecular players that are involved in here? Maybe to come up with a diagnostic. Even if you give an intervention, a typical clinical intervention will monitor for things like hemoglobin level or weight or height gain. Can we give them something more than that to wait to say that the intervention is working or not working in terms of nutrition? Finally, I want to end on a slightly different note. I feel like I'm talking to an audience that is embarking on their own scientific journeys and discoveries. And the point I want to make is about boxing yourself into categories. I think of the speakers, you've had a wide range of people here today. And I think I would caution you to be very wary of branding yourselves as biochemists, developmental biologists, neurobiologists, physicists, mathematicians. I think what's really important is to have a question that you like and to pursue it no matter where you are or what particular background you've come from. I think that we are equipped with the intelligence to tackle any problem that we set our minds to. And we should not have a fear of maths. A lot of people in my class, for example, did biology because they didn't like maths. Let me tell you, I do a lot of maths. And from those images that I showed you, I derive a lot of numbers, and I cannot make sense of any of my numbers if I didn't run particular statistics on them. And if I don't understand maths, I cannot do statistics. So I'd like to just caution students here to pursue what they feel they really like as opposed to things that sound sexy or to have a particular sort of nomenclature attached to your CV. Finally, I'd like to thank Shiva. Thank you so much for having me here. And I'd like to acknowledge Gayatri, wonderful mentor, helped me transition from the biochemist that I was to now a very pleiotropic biologist. I'm funded by the Wellcome Trust DBT India Alliance very generously with an early career fellowship. And these are all the facilities that have enabled me to do the work that I do. Thank you, and I'd be happy to take questions.
We can take a couple of questions.
Why at third instar level we have to deepen nutrition for Drosophila? Why not in first and second instar level? Any specificity?
Yes, there is a specific reason for that. Because if you deprive them of nutrition at first or second, they will never develop into pupae at all. And the whole point of this experiment was to try and make sure that they become pupae, since that's the last, that's the step. Yeah, and the third instar is the only one that will pupariate.
Humans make choices of the food that we take, right? I don't know if flies do.
They do. They have a very strong preference.
Right.
Yeah.
So you spoke about early developmental malnutrition and then good food source. Now that transition in humans is largely driven by the choice of food available to humans when they grow up. Now, how does one incorporate that element into your kind of studies? Is it possible? And are there model systems available to do that?
So, as far as I am concerned, the poor fly has no choice because they eat what I give them.
So then that's not relatable.
So intrinsically, I cannot I do not know whether they like a particular food or not. But if I have a clinical setup where somebody tells me, look, a protein to carbohydrate ratio of 2:1 seems to work very nicely in this particular intervention, I can go back and test this on the flies by changing the protein to carbohydrate ratio of the food I feed them. But will the fly prefer a McDonald's burger to a Burger King burger? I mean, I don't know.
Yeah. No, but there would be neuropeptides governing that, right? So.
Food choice?
Yes.
Food categories, perhaps, but not sure which neuropeptides they would be. There's a question there?
You talked about the size of the brain remaining same even though they are facing nutritional stress. Does that mean that the brain now has less load to take care of in the starved animal? And do you see some behavioral advantages of having a larger brain in a smaller body in a starved fly?
So it's proportionately large, but it's not big as compared to total body size, right? And the kind of study that was done here is looking at the total brain size. It's not telling you if there are more neurons or if the neurons themselves are bigger. And I think those are the type of questions I will be answering later on, but that will govern behavior basically. So just because you have less nutrition, does that mean you make less connections with 5 other neurons? I don't know.